Hello and Assalamualaikum everyone. Welcome back to our channel. Today we are going to discuss about dietary fiber, a class of food with an ugly shape but not ugly nutrition. In this video, we are going to touch a bit on what is dietary fiber, the principle of dietary fiber analysis, sample preparation, procedure, results, discussion from the experiment, and the references that we used. So, before we begin, we need to know what is dietary fiber. Dietary fiber go as a portion of ingested foodstuffs that cannot be digested by intestinal enzymes and juices of monogastric animals. It passes through the small intestine and colon undigested. It is composed of cellulose, which is the skeleton of plants, lignin, and other carbohydrates that is indigestible by members of animal species. There are two types of dietary fiber that are categorized based on their physical characteristics and effects on the body, which are insoluble fibers, cellulose, hemicellulose, lignin, and also soluble fibers, which are gum and pectin. You must be wondering how to measure fiber in our food, right? So there are several methods used for determining the dietary fiber content of foods, including proximate, gravimetric, and enzymatic gravimetric approaches. TDF, existing SDF, and IDF proportions can be determined using those methods. For example, enzymatic gravimetric method is the verified AOAC method for dietary fiber analysis. And in this video, we are going to use dates as the sample. In this experiment, 6 varieties of dates fruits are used, ranging from soft to semi-hard to hard varieties to represent the samples with significant variations in dietary fiber content, and 3 samples were taken from each variety. These are the chemicals used in this experiment, while these are the material used. Next, we need to know how the sample being processed. Firstly, samples were deseeded. The flesh was fine minced using benchtop food processor. Samples were desugared with 85% ethanol. 40 gram minced fruit was mixed with 200 ml of solvent. The fifth step is supernatant was discarded after centrifuging the mixture at 6000 GRPM for 10 minutes. The process was repeated for 5 times to eliminate the sugar. In a hot air oven at 40 degrees Celsius, the sugar date sample were dried to remove the moisture. Lastly, the final weight of the samples is to determine the bulk loss during the desugaring process. So, how dietary fiber is analyzed? The dietary fiber analysis of the desugared date samples were done by using the automated dietary fiber analyzer from NCOM Technologies. The study uses AOAC 991.43 IDF SDF method to determine the dietary fiber content. AOAC 991.43 is the enzymatic gravimetric method used to determine the IDF, SDF, and TDF content in foods involving enzymatic breakdown of protein and starch, followed by the aqueous ethanol precipitations of the fibrous components. Following that, the dietary fiber residues are weighed, and the sample's total dietary fiber content is estimated using the sample's residual protein and ash values. Each sample were observed in triplicate. Before we proceed to the most important procedure, I want to share some additional info on the stages of enzymatic digestion process. There are 3 stages of it, and the first stage is alpha amylase digestion of the samples at 95 degrees Celsius for 35 minutes. The second stage is protease digestion at 60 degrees Celsius for 30 minutes. And the third stage is sample digestion with amyloglucosidase at 60 degrees Celsius and pH between 4 to 4.5 for 30 minutes. Moving on, there are 8 steps that need to be done to complete this experiment. The first step is 0.5 gram of date samples combined with MES TRIS buffer for enzymatic digestion phase. The second step is enzymatic digestion process took place just like I've shared before. The third step is samples then filtered through IDF filter bags, and this will retain the sample's IDF components. The fourth step is the filtrate then moved to the SDF bags, where the portions were precipitated with 95% ethanol. The fifth step is mixture again filtered by STF bags to retain the remaining samples STF components. The sixth step is after the process, IDF and STF bags were gathered and washed in acetone. The seventh step is the sample components then left to dry in a hot air oven at 105 degrees Celsius overnight. And the last step is dried bags then weighed, and protein and ash contents were determined. This is the summarized results of date fruits dietary fiber analysis. To simplify the results for insoluble dietary fiber, overall, the bulk of dietary fiber in the date fruits was IDF. The Barhi variety recorded the lowest IDF content across the 6 date fruits varieties, measuring 4.63 plus minus 0.18% gram per 100 gram enzymatic methods. The highest IDF content was recorded in the Negal variety, measuring 9.69 plus minus 0.22 with the enzymatic methods. For soluble dietary fiber, the content of STF in date fruits is lower than its measured IDF content. The lowest STF content was measured in the Negal variety, measuring N0.88 plus minus 0.05% gram per 100 grams with the enzymatic methods. At the same time, the highest STF content was recorded in the Barhi variety, which measured 1.54 plus minus 0.32% gram per 100 grams with the enzymatic methods. Precisely, insoluble dietary fiber plus soluble dietary fiber will equal to total dietary fiber. So let's discuss. Date fruits popular in the Middle East are adaptable to arid climates and contain carbohydrates, soluble sugars, dietary fiber, polyphenols, vitamins and minerals, with the composition varying based on maturity, with starch degrading to glucose, fructose, and sucrose in fully mature fruits. AOAC 991.43, a modified version of AOAC 985.29, allows for the measurement of TDF, IDF, and STF using the same 3 enzymes. Alpha amylase enzyme breaks down alpha-linked polysaccharides. The protease enzyme hydrolyzes protein, while the amyloglucosidase enzyme breaks down alpha-1,4 and alpha-1,6 glycosidic bonds in starch, resulting in glucose units. Enzymatic digestion had no impact on lignin content in the date fruits, which is a significant component of insoluble dietary fiber. Most dietary fiber in date fruits is insoluble, with lignin playing a major role. The correlation between enzymatic and non-enzymatic methods for measuring total dietary fiber TDF was high, but the correlation for soluble dietary fiber STF was lower due to the original method's limitation in measuring low molecular weight STF. Only AOAC 2011.25, the only method that measures low molecular weight soluble dietary fiber STF, influenced by factors like enzyme type, purity, incubation conditions, and precipitation, leading to significant differences in STF quantification compared to other methods.